Cloud. All right. Hello. Let's talk about data replication, binary fission, and mitosis. All right. First of all, one of the key tenets in something called cell theory is that all cells divide. Okay. So no living organism can reproduce without cell division. Okay. Unicellular organisms like bacteria, one cell divides into two cells, and one organism divides into two organisms. Okay, so unicellular organisms divide in order to reproduce. Okay, for us, multicellular organisms, cell division produces a continuous supply of replacement cells. Okay, so you're literally losing millions of cells per minute. Okay, mostly off the surface of your skin, your internal, your stomach lining, etc., your hair. You're constantly losing cells. Okay, the only way you stay alive is that you have to replace the cells that you lose. Okay, and that is cell division. Okay, so here is the sexual life cycle. Okay, eukaryotic cells. We're going to start from here. The fertilized egg. Oops. The fertilized egg. All of this. Oops. I went the wrong way. Okay, sorry. From the fertilized egg to the baby to the adult you, this is all mitosis. Okay, there's something else called meiosis, which we'll cover in the next chapter. So, this is meiosis, and this only occurs in one particular place. Okay, in the testes of males and ovaries of females. Okay, in our gonads. Okay. Okay, and meiosis only occurs during fertilization. So to produce gametes and they fertilize, and we'll cover this a lot more in the next chapter. That's what next chapter is all about. Okay, so mitosis has many roles. So grow and develop, okay, repair tissues, and regenerate lost parts. Okay. All right, cells die all the time. Okay, sometimes they die because they're damaged. Sometimes they die because that's what they're supposed to do. They die. Okay, so if you look at the feet between chickens and ducks, okay, ducks have these web feet, chickens don't. And what actually happens is the feet in fetuses originally look, well, they're not fetuses, but they're embryos inside the egg, about the same. Okay, and in ducks, this membrane doesn't go away. In chickens, the membrane itself dies. Okay, and therefore you have no webbing. Okay, the same is true in humans. So, sorry, that's the head. You see between my fingers, there's a little bit of webbing, right? Just like in everybody's. Okay, but in humans, the webbing as fetuses actually goes all the way up to here. Okay, where my finger is. Okay, what happens is as you mature as a fetus, that webbing starts to die out. Okay, it undergoes this process called apoptosis. Okay, apoptosis is not just cell death, it is programmed cell death. These things are supposed to die on their own. Okay, they're programmed for that. Okay. Okay. So you see people with screwed up versions of this, like they have webbed toes and webbed fingers, and stuff like that. So webbed toes you can actually see a little bit more frequently because they usually don't fix that because they usually don't care what the toes look like too much. But webbed fingers, they have surgeries to remove the extra webbing. Okay. All right. So those liver cells contain the same DNA as his brain cells and heart cells. The only thing that's different is the sperm, because the sperm are going to be for meiosis. Okay. Okay. DNA replication is linked to cell division, so the cells must replicate their DNA before they can divide. All right. Make sure. Okay. Okay. So in DNA replication, both strands are copied simultaneously. Okay. So there is one strand here, and so let me show you what that means. There is one strand going this way, and one strand going that way. Okay. These in DNA replication, both strands are copied at the same time. Okay, so what actually happens is this. Okay, so DNA replication is semi-conservative. Okay, the darker colors are the original strands, the lighter colors are the daughter strands, the new strands. Okay, so you can see the DNA ladder literally splits open, and then you just copy both sides of the ladder simultaneously. Okay, and there are enzymes that actually replicate the DNA. Okay, and these are helicases. So helicases unwind the DNA. DNA polymerases synthesize the new strands. So, what a helicase does is actually something called unzipping. So, DNA is kind of like a zipper. You can unzip down the middle. So that's the job of the helicase. Okay. The helicase enzyme unwinds and unzips the DNA. The DNA polymerase enzyme assembles the two sides and proofreads the new strands. And then the ligases, okay, actually repair the little glitches in one side of it. The short strands joined into new strands. Okay. And of course, this uses tremendous amounts of energy. Okay, so you don't want to make you don't want to replicate your DNA unless you're planning to divide. Okay. Okay. So replication enzymes divide up the work. So DNA replication divides simultaneously in multiple spots. Okay. They're called replication bubbles, or more scientifically, they're called ORI, origins of replication. So the answer should be B, because one of them is conservative, one of them is new, and one of them is old. Okay. All right. Prokaryotes divide by binary fission. Okay. So in bacteria and eukaryotes, okay, they actually divide by something called binary fission. Since they only have one chromosome, it's actually really easy to do. You take that one chromosome, you make a copy of it, and you just kind of move one chromosome to one side and the other chromosome to the other side, and you grow a new membrane in the middle, and that's it. Binary fission is a lot easier, a lot cleaner. And it helps to have your DNA in a circle. Okay, to do that. The problem with this is that one circle cannot possibly carry that much genetic information. So you're very limited to the amount of genetic information you actually carry. Okay? All right. Okay, so prokaryotes, however, can get new DNA from other sources, like other bacteria. So they can have something called bacterial sex, basically. Okay. So they can get DNA a couple of different ways. One is something called the sex pili. Okay. So here's a sex pili that transfers genetic information from one cell to another. Okay. And if you're ever working in healthcare, you realize this is a very big problem because bacteria can transfer antibiotic resistance genes that way. Okay. The other way things work is that you can have cells dying, and as cells die, they will actually rip apart their DNA in small chunks. And what happens is when nearby cells are near pieces of DNA, they can actually uptake the new piece of DNA, especially if they're stressed. Okay, so they can grab pieces of DNA from the dying cell here. Okay, so why is binary fission? Because there's no sex involved. Okay, so now that we're talking about DNA replication, okay, after DNA replication and before cell division, okay, it has to condense, okay, it has to pack, okay. Usually, when you look at cells, their DNA is spread all over the nucleus, okay, and that's great when things are functioning, but it's not great when cells are dividing. If you imagine your room or your house, okay, you have stuff scattered everywhere. Okay, and that's the way it should be. You have pencils in places, you have like your computer in one place, you have your toilet paper near the bathroom, hopefully, right? But that's where it should be. But if you're planning on moving, you need to pack everything into a tight box so you can move it easier. And that's what the condensing is. Okay. All right. Okay. So eukaryotic chromosomes have highly condensed DNA. So that the chromosomes that you guys are used to thinking about, like if I ask you had to think of a chromosome, you think of something like an X structure. And that's only really true when the DNA is ready to replicate. When the DNA is only ready to replicate, okay, it has two identical chromosomes that attach to each other. The red and the green are identical. So you when you replicate, you put the new one right next to the old one, and so these form sister chromatids. Okay, and so this is a replicated chromosome. So again, these are so the same DNA that's in the green is in the red. Okay. Okay. So eukaryotic chromosomes are visible during cell division and only during cell division. Okay. All right. Okay, so mitosis generates exact cell copies. Okay, so mitosis is one part of the replication cycle. So let's talk about the entire replication cycle first. Okay. Okay. Ow. Okay. All right. So when cells divide, so these are cells. Okay. So cells come up through this phase. So right here, they make two cells. Okay. One cell to go away, and you're left with the other cell. Okay. That one cell is going to be small. Okay. Because it just divided itself in two. That new cell is going to be small. Okay. So what happens during G1 is mostly cell growth. Okay. So the cells will grow, get bigger, and get bigger, and get bigger. Okay. That's normal cell functioning. Okay. So it has to replace like half the stuff it just lost. Okay. And then when it's ready, okay, there's a checkpoint here that they don't show. When it's ready, it will pass this checkpoint. And after it passes checkpoint, it will undergo DNA replication. Okay, and that's the S phase. The S phase is DNA replication. Okay, when it replicates in DNA, it then goes into the G2 phase. The G2 phase is how you get slightly bigger, and that's true, but the most important thing is the preparation for cell division. Okay, in mitosis, all the DNA is packed up. So there's no more room and no more space and no more time actually to make new proteins. Okay, so in G2, it has to make all the proteins that it will need for mitosis and get everything ready before cell division. Okay. Okay. See, this G1 essays and G2 phase together are called interphase. Okay. The reason why we call it interphase is kind of archaic. When we first were able to look at cells under the microscope, we could see cells dividing, but we couldn't see very much of anything else. So we had a mitotic phase, and then the interphase was between mitotic phases. Okay. Interphase 1, SH2. Okay. Oh, cells leaving G2 phase enter mitosis, division of the nucleus, which is followed by cytokinesis. Okay, which is division of the cytoplasm. Okay. So let's talk about the series of steps. Okay, what will be most important in this quiz is going to be mitosis. Okay, mitosis and meiosis. Okay, so make sure you understand what happens. Okay, in the beginning of mitosis, the DNA condenses into chromosomes. Okay, at the same time, a spindle fiber spindle forms is made of microtubules. So you can see here, let's see if I can find a better picture. Okay, doesn't really have that great picture, but that's okay. There we go. That's the one I was looking for. Okay, so in early prophase, the chromosome condenses. Okay, and here you have that spindle fiber forming. In late prophase, the nuclear envelope breaks up. Okay, the nuclear envelope is no longer needed to keep the nucleus together because you will be pulling the DNA apart. Okay, so this happens in prophase. Okay, you can see there's a nucleus here, but there's no nucleus there left. Okay, prophase is followed by metaphase. This is metaphase. Metaphase is really easy to recognize. Okay, all the cells, all the DNA is lined up very nicely in a row right here. Okay. In anaphase, the cell starting to split apart, and it does it first by pulling the DNA to either side. So in metaphase, all the DNA is in a row and it's pulled apart. Okay, and so this is anaphase, the DNA is pulled apart. In telophase, okay, you start to form two new nuclei. Okay, and then in cytokinesis is the division of the cytoplasm. So all you do here is you form a new ring and you start to pull to either side, and the cytoplasm either goes to the left or right, and you eventually form two new cells. This is okay. In plants, cytokinesis form those things a little bit differently, but you don't need to know that. Okay, in fungi and thymos, mitosis can occur without cytokinesis. Okay, and what you get are cells that contain multiple nuclei. This happens in humans too, especially in your muscle cells. Your muscle cells are actually really long cells that have something like a couple hundred nuclei. And that's because they undergo mitosis many times without undergoing cytokinesis. Okay. Okay, so the cell cycle is under very tight control. Okay, so there are a series of chemical checkpoints that regulate the cell cycle. I already mentioned one here. There's also one here. Okay, so checkpoints ensure that all the DNA has been replicated properly, that the DNA is not damaged. There's one there as well. Okay, and the chromosomes line up in separate properties. Okay, so the DNA is not damaged, it's this one. All the DNA has been replicated, it's that one. And this goes over here. Okay, so there's 3 checkpoints. Okay, if you don't pass the checkpoint, you don't get to move on. Okay, you have to make that way you can control cell division and you can make sure everything's right before moving on. Okay, if you don't pass the checkpoint, one of two things happen. Either you stall and you fix whatever's the problem, and then you move on, or you die. Okay, the cell dies. Okay, because, and this is for the next step. Okay, cancer cells divide out of control. In cancer cells, what actually happens is that cancer cells override the checkpoint somehow. They find a way around the checkpoint and ignore them. Okay, and then the cell keeps replicating even when it shouldn't. Okay, so that's what cancer cells are. Okay, they divide out of control. And they grow into mass collagen. Okay, a tumor is either contained, in which case it's a benign tumor, which means you don't have that much to worry about, or a malignant tumor. A malignant tumor is it means that it can possibly spread. Okay. The way to treat tumors, okay, if it's benign, then you just remove it. Okay. If you can't do it, then you have, like, if it's spread all over your body, and what you get is radiation therapy or chemotherapy. Chemotherapy are just anti-cancer drugs that slow or block cell division. Okay, because these cells are divided uncontrollably. So if you can stop that from happening, then it's cancer. Okay, radiation also kills targeted tumor cells. Okay, because again they target cell division. Okay, so that's the risk factors. Okay. All right. Hopefully that makes sense to you all. I will see you guys on Wednesday.